Excellent.
Take it away.
Thank you. Hi, everybody. Ooh, it's loud. First of all, I'd like to thank the organizers for organizing such an amazing meeting every year. This is just incredible. And it's really exciting to be able to present in such a beautiful venue to friends and colleagues. My name is Yuta Lee, founder and CEO of Accelerator Biosciences. And today, my presentation will come in two parts. So I will first briefly introduce our human Trophoblast stem cell platform. And then I'll direct your attention to the cell senescent data from our collaboration with the National Institute on Aging. So Accelerator Biosciences is a stem cell platform company based in Philadelphia. We have full freedom to operate for the human trophoblast stem cell. We'll call them HTSCs. And my job is really just get them to a GMP status, which I've done in April, just a couple of months ago, and provide this platform with full regulatory package for all cell and gene therapy related companies to use as a starting stem cell source, hopefully to accelerate your therapies to market. So in cell and gene therapy today, the biggest challenge is finding the right stem cell source to start with. Right? Ethical issues, inconsistent cell source. You have to choose between scalability and immunogenicity. Many times unclear FTO and CMC documentation. We've actually taken care of all these things. Our cells present markers of all three germ layers. They're ethically sourced, scalable, immune privileged naturally, pathogen-free, have chromosomal stability to high population doublings, and we have a large IP estate to protect it. So this is where all stem cells come from. Our cells, HTSCs, are from an ectopic pregnancy sourced between 4 to 8 weeks post-fertilization, so still in the embryonic stage. And on the left side here, you'll see a blastocyst at 5 days. Our cells are from the trophectoderm lineage, the ring on the outside, which later develops into the placenta. And what happens in an ectopic pregnancy is that the embryo is traveling down a fallopian tube and it gets stuck. And it will continue to grow until it bursts the fallopian tube. And if it bursts the tube, the mother may die of internal bleeding. And so typically what happens is between 4 to 8 weeks, the mother will feel a sharp pain, go to the ER, get scanned, and the doctor will go, oops, you have a tubal ectopic pregnancy. We have to schedule a self-injectomy to take it out. And so the doctor will go in, take it out, and they will actually give it to a pathologist to look at it, confirm it, and they'll throw it away. And the reason they do that is because the embryo, or the blastocyst on the left side, needs to implant into the mother's endometrium by day 7 or day 8. But you don't discover an ectopic until 4 to 8 weeks after. So there are many weeks in between where that embryo has zero connection to the mother, no nutrients coming to it. So all OBGYNs understand that this is a non-viable fetus. And so when they take it out, all you need to do is present it to the pathologist who looks at it and they throw it away. What we've done is when they take the mass out, we scraped off the pre-placental tissue on the outside, specifically the chorionic villi. Then we hand the mass back to the pathologist who checks it and still throws it away. So it turns out then we have the earliest stem cells you can possibly source without any ethical issues, still in the embryonic stage. Okay? We have optimized our protocol to expand our cells out to 85 population doublings. So that 100 million cells per dose, that is 8 times 10 to the 17 doses. That's more cells they never need. Okay. MSCs, or mesenchymal stromal cells, by comparison, are down here. Down here around 25 population doublings, maybe 30 if you're very lucky. So at 85, we've solved the scale problem. Okay. Our cells also naturally express HLA-G. So the embryo is a combination of the father and the mother's gene. So how come this thing is able to implant it in the mother's endometrium without her immune system going completely crazy, right? Turns out this HLA-G allows for the dampening down of the mother's immune system so that the embryo can survive attached to the mother, okay? And so this is exactly the same reason why today you can actually take a fertilized egg and have a surrogate mother carry that embryo to term for you, which is incredible. So our cells, naturally from the placental origin, express this HLA-G, and when we differentiate them or skew them towards neuroprogenitor or pancreatic progenitors, they stay. So we've done 5 different animal studies already, and all with human cells and immune-competent rats, and all the cells survive. They home to the organs that we want, and no teratoma or tumor formation, and we get the behavioral improvements as well. When we did the expansion, we did the last stat at 72 population doublings, and as you can see here, the genome is very, very, very stable. We believe that the reason is because these cells have literally been incubated in the fallopian tube and have never gone to the uterus, so there's no microenvironments affecting it, and hasn't gone through term, no implantation. So very, very stable early cells. We currently have 14 different donor cell lines that are sourced in Taiwan. We've taken 1 all the way to GMP already. But because we're in Taiwan, 1/3 of the women that come in have HBV, hepatitis B, and more than 90% of them have CMV. Okay? So when you find these things out, you have to throw the cells away. Before I threw them away, though, I took the cells and sent them to IDEXX for H-Impact-1 to test for 25 human pathogens, and they come back completely clean. So that means that even though the mother may have HBV or CMV, these cells are negative for all pathogens. Right? So there's a defensive mechanism at the fetal-maternal interface that we don't fully currently understand because we can't study healthy pregnancies. So we have over 100 patents, 54 granted already, and we have taken 1 of the lines all the way to GMP, and we're providing the research use line along with the GMP line from the same donor, right? So the significance of this is that if you take the research use line and you succeed with your research, all you have to do is come back and get the GMP line, and you're off and running to IND-enabling studies, okay? So we have perfect product market fit. We have the earliest embryonic stage cells without ethical issues. It's a platform that provides scale and immunogenicity, an immune privilege, excuse me. We de-risk the FTO, the freedom to operate, with a large IP portfolio. We provide the cells with IND-ready CMC documentation, and we're headed towards a FDA drug master file by the end of the year. And we want to just provide this for all of you here, researchers, companies, to collaborate with to take your programs forward. Okay, now I'd like to direct your attention to our collaboration with the NIA. A senior researcher named Katbi Abdelmohsen at the NIA used our HTSC Secretome on a cell senescent model. So I think everybody's very familiar with this, especially after 4 days. We are focusing on cell senescence, so one of the hallmarks of aging. And the senescent cells secrete SASPs, right? And these SASP factors they promote age-related diseases such as tumor promotion and inflammation. So based on the heterochronic parabiosis studies done a while ago, the NIA was really inspired to ask, if that works, what would happen if you take the earliest stem cells that you can find and capture all the secretome and apply that on cell senescence? So that's what he did. Okay? And for those of you not familiar with exosomes or EVs, these are nanoparticles released by cells, usually between 30 to 150 nanometers. And they contain several biomolecules, proteins, DNA, RNA, and they act as messengers carrying messages from one cell to another, right? And when the EVs or exosomes are received by the recipient cells, it can change the gene expression profile of the recipient cell. So to test the effect of the stem cell secretome and exosome, our collaborators at the NIA induced senescence by using ionizing radiation on WI38 cells. And then they assessed the SASP mRNA of the conditioned media as well as the EVs, and observed that a reduction in SASP encoding our mRNAs. So the question is how? So we went through a 3-step process: characterization, in vitro, and in vivo. So how does a human trophoblast stem cell conditioned media suppress SASPs? So we use unbiased approaches to characterize the conditioned media and exosomes as well as the recipient cells. So cytokine array, bioplex, mass spec, transcriptomics, and Olink. And the cytokine array and bioplex indicated that a distinct composition in the conditioned media includes PTX3, for example, MMPs. Okay, so there are a lot of factors that are being secreted in comparison to the culture media. And mass spec analysis revealed that the composition of EBPs that includes similar contents like the cytokine array and bioplex. We also detected about 132 proteins that are enriched in the EVPs but downregulated in senescent cells. So this indicates that the potential restoration of key proteins that are otherwise downregulated in senescence. Together, these proteins might contribute to downregulation of SAS factors in senescence by the HTSC conditioned media or EVPs. We also assessed the secretome of the HTSC conditioned media by Olink, and we ID'd a large number of proteins including in the secretome of the HTSCs, including the PTX3 and MMPs. So collectively, these proteins are involved in growth, extracellular matrix, and cytokine activity, and signaling pathways. And these pathways can contribute to the suppression of SASPs. Okay, so here's the in vitro data. We treated WI38 cells with ionizing radiation to induce cell senescence and added the conditioned media. Then 5 days later, we performed the RNA-seq analysis. As you can see in the volcano plot, we observed a remarkable downregulation of several transcripts, including SASP factors such as GDF15 and CXCL1. So the left side green is downregulated, and the right side red is upregulated. And we're still looking into some of the upregulated factors. And in the previous slide, we saw, we looked at mRNA, and then we measured directly some of the secreted proteins using bioplex and found a reduction in several SASP SAS proteins, including GDF15 and CXCL1. This data indicates that the HTSC conditioned media is downregulating SAS transcripts and therefore SAS proteins. We performed Olink analysis to gain a better understanding of the whole secretome, and we observed changes in the dynamics of the secretome. Many SAS proteins are downregulated, including GDF15 and CXCL1, and we observed increased levels of other proteins, such as collagen, which is involved in extracellular matrix remodeling. So in short, HTSC secretome is downregulating what is normally up in cell senescence and upregulating what is normally down in cell senescence. And since we observed a decrease in SASP mRNA, we examined the expression of NF-kappa B and found that the phospho NF-kappa B is downregulated in cells treated with HTSC conditioned media, indicating that the conditioned media is suppressing activity of NF-kappa B, which is known to drive transcription of SAS encoding mRNAs. And here's the in vivo data. So we injected culture a conditioned media twice per week for 2 weeks and collected serum to assess aging and inflammation markers. The preliminary data indicated a decrease in GDF15, an aging marker, a CXCL1 inflammation marker, DPP4, as well as BAFF. And this was done with a dose of 100 microliters, or about 50 million particles per ml. We are now planning a larger cohort. Now with this positive data, we're planning a larger cohort and trying to determine how different doses can affect the SASP readings and control. Okay, so we found a rich composition. Well, excuse me. In summary, can early stem cell secretome have an effect on cellular senescence? And the quick answer is yes, but how? So we found rich composition in the conditioned media that could contribute to suppression of SASP, the most likely through the NF-kappa B pathway. And we also found a decrease in GDF15 and CXCL1 in vivo. So there's a lot more to do. This is a very early study. So further characterization of the HTSC EVs. We need to inject them in young and aged mice in disease models as well, and also have a further understanding of the molecular mechanisms, the epigenetic programming, reprogramming, and aging clocks, which is very exciting to me. But since the beginning of the collaboration with the NIA, we have expanded our isolation and manufacturing of EVs to a 5-liter scale in 3D, and can provide vials at 100 billion particles per ml with over 90% purity for tetraspanins. Those are markers to identify exosomes. Okay? And we'll continue to provide these EVs to the NIA for further studies. And for all of you here, if you believe that these HTSC cells or EVs will be helpful for your research or your programs, please contact me. Happy to collaborate with everyone, okay? And we're looking for collaborators specifically for the exosomes to go into longevity indications. I think that's very, very exciting. I'd like to acknowledge Dr. Kadbi Abdelmohsen, as well as Dr. Ferrucci for supporting this program at the NIH or NIA. Also Miriam Garospi, Rafa Cabo, and Isabel Bierman, who are all here at this conference. So thank you very much. And here's my QR code. So if you want to reach me, can talk to me afterwards or scan me and email me, get in touch. Thank you very much.
Thank you so much, Yuta. Great talk.
Yeah, very nice.
Thank you.
Work. I'm curious if you also looked at microRNAs, because those are being implicated in EVs.
Yes, they are. We are planning on that. So if you see in the further studies, we are mapping out, we want to map out more things. And, the first pilot study is more focused on just the conditioned media. So we want to make sure it works first, right? And now that we know there's an effect, we really want to expand that. And so within that study, we hope to go in and really look into the details of what's inside. And so that's going to take a little bit of time. So hopefully we'll be able to come back next year and present that data.
Great. More questions for Yuta? Otherwise, oh, was there one on? Yeah, go ahead.
Obviously, you're injecting quite a complex mixture when you put the conditioned media in vivo. Do you have any idea of the particular factor or factors which are, for example, downregulating GDF15?
Yeah, we actually have been thinking about, we haven't done that, but we're thinking about talking to some AI companies here to help us figure out and maybe make those links with the data that we do have. So, but that's a very interesting point, and it's a very good question, and it's absolutely a necessary thing to do, and we believe we will do that in the next cohort. Thank you.
All right, thank you so much, Yuta. That was really a great talk. Fantastic. Thank you so much.